The quiet chemistry of time, how molecules transform our world. In the gentle passage of moments that flow like streams through the landscape of existence, time reveals itself not as an abstract concept but as a patient chemist, working with molecules and atoms to transform the world through processes so subtle they often pass unnoticed. The chemistry of time operates in the browning of an apple left on a windowsill, the gradual fading of ink on treasured letters, the slow weathering of mountains into sand, and the quiet transformation of memories as they settle into the deeper layers of consciousness. This molecular dance of change encompasses every aspect of the physical world, from the rust that blooms on iron like flowers of oxidation to the complex cascade of reactions that turns the vibrant green of leaves into the golden tapestry of autumn. Time, in its chemical manifestation, becomes visible in the patina that forms on copper, the crystallization of honey, the degradation of ancient manuscripts, and the slow metamorphosis of organic matter into soil that nourishes new generations of life. The contemplation of time's chemistry invites awareness into the recognition that change is not merely an external phenomenon, but the fundamental nature of all existence. Every breath involves countless chemical transformations. Every heartbeat moves molecules through elaborate pathways. Every moment of consciousness emerges from the delicate chemistry of neural networks that themselves change with each passing second. The boundary between the observer and the observed dissolves in the recognition that both participate in the same ongoing chemical conversation with time. As consciousness settles into this gentle exploration of molecular transformation, there emerges a profound sense of participating in processes that connect the most intimate aspects of personal experience with the cosmic scale evolution of stars, planets, and galaxies. The chemistry of time operates with the same fundamental principles, whether it is creating the elements in stellar cores or facilitating the formation of new memories in the intricate architecture of the brain. In this recognition, the mind finds a rhythm that synchronizes with the patient pulse of chemical change, discovering in the molecular world a reflection of its own capacity for transformation and growth. The rust of memory. In the quiet corners of the world, where iron meets moisture and time extends its patient influence, the chemistry of oxidation unfolds as one of nature's most familiar demonstrations of temporal transformation. The rust that blooms on forgotten tools, abandoned automobiles, and weathered bridges represents more than simple decay. It embodies the fundamental principle that all matter participates in an ongoing conversation with its environment, exchanging electrons and atoms in processes that bridge the microscopic realm of molecular interaction with the visible world of form and color. The iron atom, stable in its metallic lattice for countless years, encounters water molecules carrying dissolved oxygen in an atmospheric dance that has been occurring since the Earth first developed an oxygen-rich atmosphere billions of years ago. This meeting initiates a cascade of electron transfers that transform the lustrous grey metal into the warm oranges and deep browns of iron oxide, each molecule of rust representing a story of chemical surrender and transformation that spans moments, days, or decades depending on the environmental conditions that facilitate or inhibit the reaction. The process begins at the molecular level with the dissolution of iron atoms from the metal surface, each atom releasing electrons in an oxidation reaction that occurs simultaneously with the reduction of oxygen molecules in the surrounding moisture. These complementary processes create an electrochemical circuit that operates through the thin film of water that coats most surfaces, turning every rusty object into a microscopic battery where chemical energy drives the transformation of metallic iron into hydrated iron oxides that form the characteristic rust formations. The colors that emerge during rusting tell the story of different chemical compounds forming as the reaction progresses through various stages. The initial yellow-green of iron hydroxide gives way to the darker oranges and reds of more complex iron oxide hydrates, while the deep brown and black formations indicate the presence of different crystalline structures that form as water content and environmental conditions vary over time. Each color represents a different molecular arrangement, a distinct chemical species that embodies a particular moment in the ongoing transformation of iron into rust. The texture and pattern of rust formation reflect the microscopic landscape of the original metal surface, where grain boundaries, inclusions, and stress concentrations create preferential sites for the initiation of oxidation reactions. The rust that forms follows the hidden geography of the metal, revealing through its patterns the internal structure that was invisible when the iron gleamed with metallic luster. The pitting and scaling that characterize advanced rust represent the three-dimensional architecture of chemical transformation, where new compounds expand beyond the original boundaries of the metal surface. The temporal aspect of rusting varies enormously depending on environmental factors that control the availability of water and oxygen, the presence of catalytic substances like salt that accelerate electron transfer, and the protective or corrosive nature of surrounding materials. A tool stored in dry air may show no rust after decades, while the same tool exposed to marine environments can transform completely within months, demonstrating how environmental chemistry controls the rate at which time's transformations manifest in the physical world. The beauty that emerges from rust formation speaks to the aesthetic dimension of chemical change, where the ordered decay of metallic surfaces creates patterns and colors that rival those found in intentional art. The flowing forms of rust stains on concrete, the delicate lace-like patterns of corrosion on thin metal sheets, and the rich earth tones of aged iron create visual compositions that emerge from the intersection of chemistry and time, revealing the artistic potential inherent in molecular transformation. The symbolic resonance of rust extends beyond its chemical reality to encompass metaphorical associations with memory, aging, and the passage of time in human experience. The rust that accumulates on childhood bicycles or garden tools becomes a visual marker of time's passage, while the preservation or prevention of rust represents humanity's ongoing negotiation with entropy and change. The patina that forms on copper and bronze carries similar symbolic weight, representing the beautiful aspects of aging that emerge when chemical transformation enhances rather than diminishes aesthetic value. The relationship between rust and memory operates on multiple levels, from the literal preservation of objects that carry personal or cultural significance to the metaphorical connections between chemical change and the transformation of memory over time. Just as iron slowly oxidizes through its interaction with the environment, memories undergo their own chemical transformations within neural networks, changing subtly with each recall and gradually acquiring new associations and emotional colorations that reflect the ongoing chemistry of consciousness. The prevention and treatment of rust represent humanity's attempt to control the chemical conversation between materials and time, using protective coatings, controlled atmospheres, and chemical inhibitors to slow or prevent the oxidation reactions that would otherwise transform metallic objects. These interventions demonstrate the possibility of influencing temporal chemistry while acknowledging the fundamental tendency of all systems to evolve toward greater entropy and chemical equilibrium with their surroundings. The cycle of rust formation and removal that characterizes human interaction with iron objects creates a rhythm of preservation and renewal that mirrors larger patterns of construction and decay, creation and destruction that characterize both natural and cultural evolution. The tools that rust and are restored, the bridges that corrode and are repaired, and the artworks that age and are conserved all participate in ongoing cycles of chemical transformation that connect human activity with the broader patterns of temporal change that govern all material existence. Now, close the eyes and sense the gentle process of oxidation, feeling the patient exchange of electrons between iron and oxygen, witnessing the slow blooming of rust like flowers of time opening across metallic surfaces. The chemistry of ripening, in the quiet transformation that occurs within the cellular architecture of fruits and vegetables, time expresses itself through enzyme-catalyzed reactions that convert starches to sugars, break down chlorophyll to reveal hidden pigments, and soften rigid cell walls through the action of proteins that have evolved to orchestrate the complex chemistry of ripening. The banana that changes from green to yellow to brown tells the story of time working through biological systems, creating changes that serve both the reproductive strategies of plants and the nutritional needs of animals in a choreography of chemical cooperation that spans multiple timescales and biological purposes. The green banana contains chlorophyll molecules that capture light energy for photosynthesis while the fruit develops. But as ripening begins, enzymes called chlorophyllices begin breaking down these green pigments to reveal the yellow carotenoids that were present all along but masked by the dominant chlorophyll absorption. This color change signals not merely aesthetic transformation but fundamental alterations in cellular chemistry, as the fruit transitions from an organ of photosynthesis to a structure optimized for seed dispersal through animal consumption. The conversion of starch to sugar that characterizes banana ripening involves the enzyme amylase, which breaks the long chains of glucose molecules that comprise starch into the shorter chains and individual glucose and fructose molecules that create the sweet taste associated with ripe fruit. This transformation increases not only palatability, but also the bioavailability of energy stored within the fruit, making the nutrients more accessible to both the developing seeds and the animals that will consume the fruit and disperse the seeds to new locations. The softening of fruit texture during ripening results from the action of pectinase enzymes that break down the pectin molecules in cell walls, causing the rigid structure that maintains fruit shape to gradually dissolve into a softer matrix that yields to gentle pressure. This texture change serves multiple biological functions, making the fruit easier to consume while simultaneously signaling optimal nutritional content and reducing the energy required for digestion by fruit-eating animals. The production of volatile compounds that create the characteristic aroma of ripe fruits involves complex biochemical pathways that convert amino acids, fatty acids, and other cellular components into the esters, aldehydes, and other organic molecules that stimulate olfactory receptors and create the sensory experience of fruit flavor. These aromatic compounds serve as long-distance chemical signals that advertise the fruit's readiness for consumption to animals whose digestive systems can process the seeds and transport them to suitable growing locations. The browning that occurs in overripe fruit represents a different set of chemical reactions, primarily the oxidation of phenolic compounds by enzymes called polyphenol oxidases, when cellular compartments break down and previously separated chemicals come into contact with each other and with atmospheric oxygen. This browning reaction, while often considered undesirable in stored fruit, represents part of the natural decomposition process that returns organic matter to the soil where it can nourish new plant growth. The timing of ripening involves complex hormonal signals, particularly the plant hormone ethylene, which coordinates the expression of genes responsible for producing the enzymes that drive ripening chemistry. Ethylene acts as both an internal signal within individual fruits and a communication molecule between fruits, explaining why the presence of one ripe fruit can accelerate the ripening of nearby fruit through the atmospheric transmission of this gaseous hormone. The diversity of ripening patterns among different fruits reflects the varied ecological strategies that plants have evolved for seed dispersal, with some fruits ripening quickly and synchronously to create intense but brief periods of attractiveness to dispersers, while others ripen gradually to extend the period of availability and increase the probability of successful seed dispersal across different environmental conditions and animal populations. The relationship between ripening chemistry and nutritional content demonstrates the sophisticated ways that plants optimize the timing of consumption to maximize reproductive success while providing nutritional benefits to dispersing animals. The peak vitamin content, sugar concentration, and antioxidant levels that characterize properly ripened fruit represent the culmination of months of biochemical investment in creating packages of nutrition that serve the mutual interests of plants and animals. The cultural significance of fruit ripening extends beyond biological function to encompass human relationships with agricultural timing, seasonal availability, and the aesthetic appreciation of natural change. The monitoring of fruit ripeness for optimal harvest timing, the development of techniques for controlling ripening rates, and the culinary traditions that celebrate the flavors of perfectly ripened fruit all reflect humanity's deep engagement with the temporal chemistry of plant biology. The preservation techniques that humans have developed to extend the availability of ripe fruit from controlled atmosphere storage that slows ripening chemistry to fermentation processes that transform fruit sugars into alcohol or organic acids demonstrate sophisticated understanding of how environmental conditions influence biochemical reaction rates and how these reactions can be controlled or redirected to serve human purposes while working with rather than against natural chemical processes. The metaphorical associations between fruit ripening and human development, creativity, and wisdom reflect the intuitive recognition that optimal outcomes often require patience and proper timing, that the rush to premature completion often produces inferior results, and that the natural chemistry of development contains its own wisdom about the appropriate pace of transformation and the conditions necessary for achieving full potential. The fading of colors. In the gentle dissolution of pigments that once blazed with vibrant intensity, time reveals its power to transform the visual world through photochemical reactions that break apart the molecular structures responsible for color, creating the slow fade from brilliant hues to muted tones that characterizes everything from sun-bleached fabrics to aged paintings. The chemistry of fading operates through the interaction between light energy and organic molecules, where photons carry sufficient energy to break chemical bonds and rearrange molecular structures in ways that alter or eliminate their capacity to absorb and reflect specific wavelengths of light. The fading of a red rose involves the gradual breakdown of anthocyanin molecules, complex organic compounds that absorb green and yellow light, while reflecting the red wavelengths that create the flower's characteristic color. When ultraviolet radiation from sunlight strikes these pigment molecules, it provides enough energy to break the conjugated double bonds that are responsible for the molecule's light absorption properties, transforming the colored compound into colorless degradation products that no longer contribute to the visual appearance of the flower. The process of photodegradation affects different colors at different rates, with blue and violet pigments generally being more susceptible to light-induced breakdown than red and yellow compounds, explaining why ancient textiles and artworks often show a characteristic shift toward warmer tones as the cooler pigments fade preferentially over time. This selective fading creates color changes that may be subtle initially, but become increasingly pronounced as exposure to light continues over months, years, or centuries. The interaction between oxygen and light accelerates many fading processes through photooxidation reactions, where the combination of photon energy and molecular oxygen creates highly reactive species that attack pigment molecules more aggressively than light alone. This explains why materials stored in vacuum or inert atmospheres maintain their colors longer than those exposed to air, and why antioxidant compounds can sometimes slow the fading process by intercepting the reactive oxygen species before they can damage color-producing molecules. The fading of printed materials involves the breakdown of synthetic dyes and inks, many of which were designed for visual impact rather than long-term stability, creating documents and photographs that gradually lose their chromatic intensity as the organic molecules responsible for color undergo slow chemical decomposition. The development of archival quality inks and papers represents humanity's attempt to slow these natural processes through the selection of more stable chemical compounds and protective storage conditions. The temperature dependence of fading reactions demonstrates the fundamental relationship between molecular motion and chemical reaction rates, with higher temperatures providing more thermal energy to overcome activation barriers and accelerate the breakdown of pigment molecules. This explains why materials stored in cool, dark conditions maintain their colors longer than those exposed to warm, bright environments. and why seasonal variations in temperature and light exposure can create cyclical patterns of color change. The role of moisture in fading processes adds another layer of complexity to color degradation, as water molecules can participate directly in hydrolysis, reactions that break down pigments, while also facilitating the transport of oxygen and other reactive species to sites where color molecules are located. The control of humidity therefore becomes crucial for the preservation of colored materials, requiring the maintenance of moisture levels that prevent both the excessive dryness that can make materials brittle and the high humidity that accelerates chemical degradation. The development of color-stable materials involves the synthesis of pigments and dyes with molecular structures that resist photochemical breakdown, often through the incorporation of aromatic ring systems that can dissipate absorbed light energy as heat rather than using it to drive destructive chemical reactions. These light-fast compounds represent the application of chemical knowledge to the creation of materials that can maintain their visual properties despite prolonged exposure to environmental stresses. The aesthetic dimension of color fading encompasses both the loss of original chromatic intensity and the emergence of new color relationships as different pigments fade at different rates, creating subtle shifts in hue and saturation that can sometimes enhance rather than diminish the beauty of aged materials. The patina of age that characterizes antique textiles, vintage photographs, and historical artworks often derives its appeal from these gentle color transformations that occur through the chemistry of time. The psychological impact of color fading connects the chemistry of pigment degradation with human emotional responses to change and loss, as the gradual disappearance of vibrant colors can evoke feelings of nostalgia, melancholy, or acceptance of temporal passage. The faded photograph that triggers memories of past events, the sun-bleached fabric that recalls summer days, and the aged painting that speaks of historical continuity, all demonstrate how chemical changes in color can resonate with the emotional chemistry of human experience. The preservation of colors through conservation techniques represents a form of chemical intervention in natural processes using controlled environments, protective filters, and stabilizing treatments to slow the rate of photochemical degradation, while acknowledging that complete prevention of change is neither possible nor always desirable. The goal becomes managing rather than eliminating the chemistry of time, allowing for the natural evolution of materials while preventing catastrophic degradation that would destroy cultural and personal significance. The cyclical relationship between fading and renewal that characterizes many natural systems, from the seasonal color changes of deciduous leaves to the regeneration of pigments in living systems, demonstrates that color loss is often part of larger patterns of chemical recycling and renewal rather than simple deterioration. The brown leaves that have lost their chlorophyll contribute to soil formation that nourishes new growth, while the faded flower petals provide nutrients for seed development, illustrating how the chemistry of fading serves larger purposes in the ongoing cycles of natural transformation. If this gentle vision of the cosmos soothes the mind, consider resting here more often. Subscribe to the channel and share your thoughts in the comments, a quiet echo beneath the stars. The alchemy of scent. In the invisible realm where molecules drift through air to encounter the sensitive chemistry of olfactory receptors, time manifests through the gradual evaporation and chemical transformation of fragrant compounds that carry information about the molecular world into consciousness through pathways more ancient and direct than those of sight or sound. The perfume that fades from skin, the aroma that escapes from flowers, and the scent that lingers in familiar spaces all participate in temporal chemistry that connects the microscopic behavior of volatile organic molecules with the complex neurochemistry of memory and emotion. The evaporation of perfume from skin involves the transition of aromatic molecules from liquid to gaseous states at temperatures far below their boiling points, a process driven by the kinetic energy of individual molecules that occasionally acquire sufficient velocity to break free from the attractive forces that bind them to the liquid phase. The rate of this evaporation depends on molecular size, chemical structure, and environmental conditions creating the characteristic evolution of fragrance over time as different aromatic compounds volatilize at different rates. The top notes of a perfume, composed of small, highly volatile molecules like citrus terpenes and light esters, evaporate within minutes to hours of application, creating the initial olfactory impression that quickly gives way to the heart notes of medium volatility that may persist for several hours before yielding to the base notes of larger, more stable molecules that can remain detectable for days or even weeks, depending on the specific chemical structures involved and the environmental conditions that affect evaporation rates. The chemical transformation of aromatic molecules during their residence on skin involves interactions with the slightly acidic pH of human perspiration, the enzymes present in skin cells, and the oxidizing effects of atmospheric oxygen, all of which can modify the molecular structure of fragrant compounds and alter their olfactory properties. These skin chemistry effects explain why the same perfume can smell different on different people and why fragrances continue to evolve even after the initial evaporation of the most volatile components. The natural production of aromatic compounds by flowers represents one of nature's most sophisticated applications of temporal chemistry, with plants timing the release of specific volatile molecules to coincide with the activity patterns of pollinating insects and adjusting the chemical composition of their fragrances throughout the day to optimize the probability of successful pollination. The jasmine that releases its most intense fragrance during evening hours, and the roses that produce different aromatic profiles in morning versus afternoon demonstrate how temporal chemistry serves biological communication functions. The degradation of aromatic compounds through oxidation, hydrolysis, and other chemical reactions creates the phenomenon of scent aging, where the fresh, bright character of new fragrances gradually transforms into deeper, more complex profiles as reactive molecules undergo chemical changes that produce new aromatic compounds while eliminating others. This aging process can enhance or diminish the overall attractiveness of a fragrance, depending on whether the degradation products complement or conflict with the original aromatic design. The interaction between fragrant molecules and the aqueous environment of the nasal cavity involves the dissolution of hydrophobic aromatic compounds in the mucus layer that covers olfactory receptor cells, a process that requires the molecules to partition between air and water phases in ways that make them accessible to the protein receptors that initiate the neurochemical cascade, leading to scent perception. The efficiency of this dissolution process affects both the intensity and duration of olfactory sensations. The neurochemistry of scent processing involves the conversion of molecular recognition events at olfactory receptors into patterns of electrical activity that travel directly to brain regions associated with emotion and memory, creating the intimate connection between fragrance and psychological experience that makes scent such a powerful trigger for memory retrieval and emotional response. The chemical specificity of this process allows individual aromatic molecules to evoke highly specific memories and associations. The temporal aspects of scent memory involve the formation and retrieval of associations between specific aromatic molecules and the circumstances of their original encounter, creating olfactory memories that can remain vivid and emotionally resonant for decades, despite the chemical instability of the original aromatic stimuli. The perfume that recalls a beloved person, the flower fragrance that evokes childhood experiences, and the food aromas that trigger cultural memories all demonstrate how temporal chemistry creates lasting impressions in consciousness. The preservation of fragrant materials through techniques like enfleurage, distillation, and solvent extraction represents humanity's attempt to capture and stabilize the temporal chemistry of natural aromas, creating concentrated essences that can reproduce the olfactory experience of fresh flowers, herbs, and other aromatic sources long after the original sources have undergone the chemical changes that eliminate their natural fragrance. The synthetic reproduction of natural fragrances involves the identification and chemical synthesis of the specific molecular structures responsible for desired olfactory effects. creating artificial aromatic compounds that can mimic natural scents while often providing greater stability and consistency than the chemically complex mixtures found in natural sources. This synthetic approach demonstrates advanced understanding of the relationship between molecular structure and olfactory perception. The cultural significance of fragrance, preservation, and use reflects the deep human appreciation for the temporal chemistry of scent, from the religious use of incense that creates aromatic environments for spiritual practice, to the personal use of perfumes that enhance individual identity and social interaction. The rituals surrounding fragrance application and the traditions of aromatic gift-giving demonstrate how scent chemistry becomes integrated into social and cultural practices. The environmental chemistry of scent dispersal involves the transport of aromatic molecules through air currents, their interaction with atmospheric pollutants, and their eventual degradation through photochemical reactions and other atmospheric processes that determine how far and how long natural and artificial fragrances can travel from their sources. Understanding these processes has become important for managing the environmental impact of synthetic fragrances and maintaining air quality in urban environments. Now, breathe deeply and sense the invisible chemistry of aroma, feeling the gentle drift of molecules from their sources into awareness, witnessing the temporal dance of evaporation and transformation that carries the essence of the world into consciousness. The slow dance of crystallization in the patient realm, where dissolved substances gradually organize themselves into ordered structures, time reveals its architectural powers through the crystallization processes that transform liquid solutions into solid geometries of remarkable beauty and precision. The salt that forms when seawater evaporates, the sugar that crystallizes in cooling syrup, and the minerals that grow in geological cavities all demonstrate how time and chemistry collaborate to create the structured matter that forms the foundation of both biological and inorganic organization. The nucleation that initiates crystal formation represents one of chemistry's most delicate threshold phenomena, where dissolved molecules must overcome the energy barrier required to organize into the first tiny clusters that provide templates for subsequent growth. This process requires supersaturation conditions where the concentration of dissolved material exceeds the normal solubility limit, creating thermodynamic instability that drives the transition from disordered solution to ordered solid phase. The growth of crystals proceeds through the sequential addition of molecules to the expanding solid structure, with each new molecular arrival finding its appropriate position within the existing crystal lattice, according to the geometric constraints imposed by intermolecular forces and the minimization of system energy. The regular, repeating patterns that characterize crystalline structures emerge from these local optimization processes, occurring simultaneously at countless growth sites across the crystal surface. The morphology of growing crystals reflects the balance between thermodynamic factors that favor the most stable crystal faces and kinetic factors that determine the relative growth rates of different crystal orientations under specific environmental conditions. Temperature, concentration gradients, fluid flow patterns, and the presence of impurities all influence crystal shape by affecting the local chemical environment at different parts of the growing crystal surface. The timescales involved in crystallization vary enormously depending on the chemical system and environmental conditions, from the rapid crystallization of sodium chloride from evaporating seawater that can occur within hours or days to the geological formation of large quartz crystals that requires thousands or millions of years of stable conditions. These different temporal scales demonstrate how the same fundamental processes can produce dramatically different results when operating under different environmental constraints. The role of defects in crystal growth reveals how imperfections can enhance rather than diminish the beauty of crystalline structures, with inclusions, twinning, and other irregularities creating optical effects, color variations, and structural patterns that make each crystal unique despite the underlying geometric regularity of the crystal lattice. These defects often record the history of changing conditions during crystal growth, preserving information about past environmental states. The dissolution and recrystallization processes that occur when crystals encounter changing chemical conditions demonstrate the dynamic equilibrium that exists between solid and dissolved phases, with crystal surfaces continuously exchanging molecules with surrounding solutions at rates that depend on local chemical potentials and surface energies. This ongoing molecular exchange means that even apparently stable crystals participate in constant chemical communication with their environment. The optical properties of crystals, including their characteristic colors, transparency, and light-bending effects, emerge from the interaction between electromagnetic radiation and the ordered molecular arrangements within the crystal lattice. The way crystals split light into component colors, create double images, or exhibit fluorescence under ultraviolet illumination all reflect the quantum mechanical properties of electrons within the structured chemical environment of the crystalline solid. The formation of crystal twins and other complex growth patterns demonstrates how crystallization can produce structures that transcend simple geometric regularity to create forms of surprising complexity and beauty. The interpenetration of multiple crystal orientations, the development of dendritic growth patterns, and the formation of spherolytic structures all show how local variations in growth conditions can lead to emergent complexity in crystalline architecture. The biological aspects of crystallization include both the formation of mineral structures within living organisms, such as the calcium carbonate shells of marine animals and the silica structures of diatoms, and the crystallization of biological molecules like proteins and DNA that enables their study through X-ray crystallography. These biological crystallization processes demonstrate how living systems have evolved to control and direct crystal formation for structural and functional purposes. The industrial applications of controlled crystallization encompass the production of everything from pharmaceutical compounds and electronic materials to food products and decorative objects, demonstrating humanity's growing ability to direct crystallization processes to produce materials with specific properties and characteristics. The development of techniques for controlling crystal size, shape, purity, and defect structure represents sophisticated manipulation of temporal chemistry. The preservation and collection of natural crystals reflects both scientific interest in understanding crystallization processes and aesthetic appreciation for the beauty that emerges from the intersection of chemistry and time. The formation of crystal collections, the study of mineral specimens, and the use of crystals in decorative and spiritual practices all demonstrate human fascination with the ordered structures that nature creates through patient chemical processes. The metaphorical associations between crystallization and human development, including concepts of clarity, structure, perfection, and gradual refinement, reflect intuitive recognition of parallels between chemical organization and psychological growth. The idea that wisdom crystallizes from experience, that understanding emerges from the gradual organization of knowledge, and that character develops through the slow accumulation of consistent choices all draw upon the imagery of crystallization to describe temporal processes in human experience. The chemistry of memory formation. In the intricate neural networks where experience transforms into lasting memory, time operates through biochemical processes that encode, consolidate, and retrieve information using molecular mechanisms that bridge the gap between transient electrical activity and permanent structural changes in brain tissue. The formation of memories involves cascades of chemical reactions that strengthen synaptic connections, modify protein synthesis, and create the physical substrates that allow conscious experience to leave lasting traces in the architecture of the nervous system. The initial encoding of memory begins with the release of neurotransmitter molecules at synapses, where chemical signals cross the gaps between neurons and bind to receptor proteins that initiate changes in the electrical properties of the receiving cell. These synaptic events, lasting only milliseconds, trigger longer-lasting biochemical changes that can persist for hours, days, or even lifetimes, demonstrating how brief chemical events can create enduring modifications in neural function. The process of long-term potentiation represents one of the fundamental mechanisms through which temporary electrical activity becomes converted into permanent structural change, involving the activation of protein kinases that modify existing synaptic proteins while also triggering the synthesis of new proteins that strengthen and stabilize synaptic connections. This molecular cascade demonstrates how the chemistry of memory formation operates across multiple temporal scales simultaneously. The role of calcium ions in memory formation illustrates how simple inorganic molecules can serve as crucial signals in complex biological processes, with calcium influx through specialized ion channels, triggering the activation of calcium-dependent enzymes that modify synaptic proteins and initiate the gene expression changes necessary for long-term memory storage. The concentration and timing of calcium signals help determine which synaptic changes become permanent and which fade away. The consolidation of memories involves the gradual transfer of information from temporary storage systems that depend on ongoing electrical activity to more stable storage systems that rely on structural modifications in neural connectivity. This consolidation process can take hours to years to complete and involves repeated reactivation of memory traces during sleep and quiet waking states, suggesting that memory formation continues long after the original learning experience. The molecular basis of memory retrieval involves the recreation of patterns of synaptic activity that approximate those present during the original encoding experience, with chemical signals propagating through neural networks that have been modified by previous learning to make certain activity patterns more likely to occur. The fidelity of this recreation process determines the accuracy and vividness of remembered experience. The chemical modifications that occur during memory formation include changes in the phosphorylation states of synaptic proteins, alterations in the expression levels of specific genes, and structural modifications in the shapes and connections of individual neurons. These molecular changes create the physical basis for memory storage, while also making the neural networks more responsive to similar patterns of activity in the future. The interaction between memory formation and emotional processing involves specialized neurotransmitter systems, particularly those involving dopamine, norepinephrine, and acetylcholine, that modulate the strength and persistence of memory encoding based on the emotional significance of the experience being encoded. This chemical modulation explains why emotionally significant events tend to be remembered more vividly and for longer periods than emotionally neutral experiences. The degradation and forgetting of memories involves active biochemical processes that weaken synaptic connections and remove the molecular modifications that maintain memory traces, demonstrating that forgetting is not simply the passive decay of memory traces, but an active process that helps maintain the efficiency and capacity of memory systems by eliminating outdated or irrelevant information. The formation of false memories demonstrates how the reconstructive nature of memory retrieval can lead to the incorporation of new information into existing memory traces, with the molecular mechanisms of memory modification being activated during retrieval and allowing new chemical changes to alter the stored information. This process shows how memory formation continues throughout the lifetime of a memory trace. The chemical basis of memory disorders involves disruptions in the normal biochemical processes of memory formation, consolidation, or retrieval, with conditions like Alzheimer's disease involving the accumulation of abnormal proteins that interfere with synaptic function, while other memory disorders result from genetic variations that affect the enzymes and signaling pathways involved in memory processing. The pharmacological enhancement and modification of memory involves the use of drugs that affect the neurotransmitter systems and molecular pathways involved in memory formation, demonstrating how understanding the chemistry of memory can lead to interventions that improve memory performance or reduce the impact of traumatic memories by targeting specific aspects of the memory formation process. The temporal dynamics of memory formation reveal how different types of memories form through distinct biochemical pathways operating on different timescales, with procedural memories, episodic memories, and semantic memories, each involving specialized neural circuits and molecular mechanisms that determine their characteristic properties and longevity. Understanding these differences provides insight into the diverse ways that experience can be preserved in neural chemistry. The relationship between memory chemistry and consciousness involves the recognition that the subjective experience of remembering emerges from the same molecular processes that create the objective changes in neural connectivity, suggesting that the chemistry of memory formation provides a bridge between the physical and experiential aspects of mental life through mechanisms that remain largely mysterious despite intensive scientific investigation. If this gentle vision of the cosmos soothes the mind, consider resting here more often. Subscribe to the channel and share your thoughts in the comments, a quiet echo beneath the stars. The weathering of stone. In the patient dissolution of mountains and the gradual creation of soil, time reveals its power to transform the most durable materials through chemical processes that operate across geological timescales to reshape the Earth's surface and create the conditions necessary for life. The weathering of stone involves multiple chemical mechanisms working simultaneously to break down crystalline structures, release mineral nutrients, and convert solid rock into the particles and dissolved substances that form the foundation of terrestrial ecosystems. The chemical weathering of granite begins with the slow hydrolysis of feldspar minerals, where water molecules react with aluminum silicate crystals to produce clay minerals and dissolved ions, gradually weakening the rock's structure and creating the conditions for physical breakdown. This process occurs grain by grain, crystal by crystal, as water penetrates along crystal boundaries and slowly dissolves the chemical bonds that hold the rock together. The formation of carbonic acid from atmospheric carbon dioxide dissolved in rainwater creates a weak but persistent acidic solution that accelerates the chemical breakdown of many rock types, particularly limestone and marble, where the acid reacts with calcium carbonate to produce dissolved calcium and bicarbonate ions that are carried away by groundwater and streams. This carbonation process has been operating for billions of years to shape landscape features and regulate atmospheric carbon dioxide levels. The oxidation of iron-bearing minerals in rocks creates the characteristic red and yellow coloration of weathered surfaces, while simultaneously weakening the rock structure through volume changes that occur when iron converts from its reduced to oxidized forms. The rusting of iron in rocks follows the same basic chemistry as the rusting of metallic iron, but occurs much more slowly due to the lower concentrations and reduced mobility of reactants in the solid state. The biological acceleration of rock weathering involves the production of organic acids by plant roots, bacteria, and fungi that can dissolve minerals more effectively than purely inorganic processes, while the physical penetration of roots into rock cracks creates mechanical forces that complement chemical dissolution. The symbiotic relationship between biological and geological processes demonstrates how life and Earth systems co-evolve over geological timescales. The formation of soil from weathered rock involves the accumulation of organic matter, the development of distinct layers with different chemical and physical properties, and the establishment of biological communities that further modify the chemical environment through their metabolic activities. This soil formation process can take hundreds to thousands of years to produce mature soil profiles that support complex ecosystems. The transport of weathering products by water creates the sedimentary processes that redistribute earth materials from upland areas to lowland basins, with dissolved minerals carried in solution, while solid particles are transported as suspended sediment or bedload, depending on their size and the energy of the transporting water. These transport processes connect weathering in one location with deposition in another, creating landscape-scale material cycles. The chemical composition of natural waters reflects the weathering processes occurring in their watersheds, with different rock types producing characteristic dissolved ion signatures that can be used to trace water sources and understand regional weathering patterns. The chemistry of rivers, lakes, and groundwater tells the story of rock-water interactions occurring over thousands of years as water slowly dissolves the landscape and carries the products of weathering toward the sea. The formation of secondary minerals during weathering processes demonstrates how destruction and creation occur simultaneously in geological systems, with the breakdown of primary rock-forming minerals providing the raw materials for the synthesis of new mineral phases that are more stable under surface conditions. Clay minerals, iron oxides, and other weathering products represent new chemical compounds created through the intersection of rock chemistry and atmospheric chemistry. The role of climate in controlling weathering rates reveals how temperature and moisture availability determine the speed of chemical reactions that break down rocks, with warm, humid climates promoting rapid weathering while cold, dry climates slow these processes to near standstill. The variation in weathering rates with climate helps explain the global distribution of soil types and landscape features. The preservation of ancient weathering profiles in the geological record provides evidence for past climate conditions and demonstrates how weathering processes have changed over geological time as atmospheric composition and global temperatures have varied. These paleosols, or ancient soils, preserve chemical signatures of weathering environments that existed millions of years ago. The impact of human activities on weathering processes includes the acceleration of chemical breakdown through acid rain, the exposure of fresh rock surfaces through mining and construction, and the modification of natural drainage patterns that affect how water interacts with rock and soil. These anthropogenic effects demonstrate how human activities can alter geological processes that normally operate on much longer timescales. The economic significance of weathering processes includes both the beneficial effects of soil formation and mineral concentration and the detrimental effects of building deterioration and infrastructure damage. The same chemical processes that create fertile soils also attack concrete, corrode metal structures, and weaken rock foundations, demonstrating how geological timescale processes affect human timescale concerns. The aesthetic appreciation of weathered landscapes reflects human recognition of the beauty that emerges from the slow transformation of rock into sculptured forms, colored surfaces, and textured patterns that record the history of chemical interaction between earth materials and atmospheric conditions. The patina of age on stone buildings, the carved forms of wind and water erosion, and the rich colors of oxidized rock surfaces all demonstrate the artistic potential of geological chemistry. The philosophical implications of rock weathering encompass themes of persistence and change, durability and fragility, and the relationship between geological and human timescales. The recognition that even the most durable materials eventually succumb to chemical breakdown provides perspective on the temporal nature of all material existence, while demonstrating the creative potential inherent in processes of dissolution and transformation. Now, feel the patient work of water and air upon stone, sensing the slow chemical conversation between rock and atmosphere that has been shaping the earth for billions of years. Witnessing the transformation of mountains into soil through chemistry, so gradual it spans countless human generations. The molecular dance of decay. In the gentle return of organic matter to its elemental components, time orchestrates the complex biochemical processes of decomposition that transform the structures of life into the raw materials for new growth. The fallen leaf that crumbles into humus, the wooden post that slowly returns to soil, and the cycle of death and renewal that characterizes all biological systems demonstrate how time operates through enzymatic chemistry to maintain the circulation of elements essential for life. The initial stages of decomposition begin even before visible decay becomes apparent, as cellular enzymes released by the breakdown of cell membranes begin to hydrolyze proteins, carbohydrates, and other biological molecules into smaller fragments that can be metabolized by decomposer organisms. This autolysis represents the beginning of the chemical cascade that will eventually reduce complex biological structures to simple inorganic compounds. The colonization of dead organic matter by bacteria and fungi introduces specialized enzyme systems that can break down the structural components of biological tissues, including cellulose, lignin, and chitin that resist breakdown by the enzymes originally present in living cells. These decomposer organisms secrete extracellular enzymes that digest complex polymers outside their cells before absorbing the resulting small molecules for their own metabolic needs. The chemistry of lignin decomposition represents one of nature's most challenging biochemical problems, as this complex polymer that provides strength and rigidity to woody tissues requires specialized enzyme systems that can cleave the irregular bonds connecting its aromatic subunits. The white rot fungi that accomplish this decomposition use oxidative enzymes that generate highly reactive radicals capable of breaking down lignin's recalcitrant structure. The temporal sequence of decomposition reflects the varying resistance of different biological molecules to enzymatic breakdown, with readily digestible compounds like sugars and amino acids disappearing first, followed by more complex materials like cellulose and proteins, and finally by the most resistant components like lignin and waxy compounds that may persist for years or decades under favorable preservation conditions. The environmental factors that control decomposition rates include temperature, moisture, oxygen availability, and pH, all of which affect both the activity of decomposer enzymes and the growth rates of the microorganisms that produce them. The preservation of organic matter in cold, dry, or acidic conditions demonstrates how environmental chemistry can slow or halt the normal processes of biological decomposition. The formation of humus from decomposed organic matter involves the chemical condensation of partially degraded biological molecules into complex, dark-colored polymers that resist further decomposition and provide stable organic matter in soils. This humification process creates organic compounds that can persist in soils for hundreds or thousands of years while slowly releasing nutrients for plant growth. The nitrogen cycle that operates during decomposition involves the conversion of organic nitrogen in proteins and nucleic acids into ammonia, nitrites, and nitrates through the sequential action of different bacterial enzyme systems. This microbial processing makes nitrogen from dead organisms available for uptake by living plants, demonstrating how decomposition chemistry supports the continued productivity of ecosystems. The production of gases during decomposition, including carbon dioxide, methane, hydrogen sulfide, and various organic compounds, creates the characteristic odors associated with decay, while also contributing to atmospheric chemistry and global biogeochemical cycles. The anaerobic decomposition that occurs in waterlogged soils and sediments produces different gas mixtures than aerobic decomposition, reflecting the different biochemical pathways available under different oxygen conditions. The role of decomposition in carbon cycling demonstrates how the chemistry of decay regulates the global carbon balance by determining how quickly carbon fixed in biological tissues returns to atmospheric carbon dioxide. The residence time of carbon in different types of organic matter, from rapidly decomposing leaves to slowly decaying wood, affects both local soil carbon storage and global climate regulation. The chemical preservation of organic matter under special conditions creates fossil deposits that preserve evidence of ancient life forms and past environmental conditions. The formation of coal, oil, and other fossil fuels represents incomplete decomposition under anaerobic conditions, while the preservation of organic molecules in amber, ice, and other protective media provides direct chemical evidence of past biological processes. The human management of decomposition processes includes composting techniques that optimize conditions for rapid, controlled decay of organic waste, preservation methods that slow decomposition of food and other materials, and understanding of how decomposition affects the stability and safety of organic materials in various applications. These applications demonstrate how knowledge of decomposition chemistry can be used to enhance beneficial processes while preventing unwanted decay. The aesthetic and cultural significance of decomposition encompasses both the acceptance of decay as a natural process essential for renewal and the human tendency to resist or disguise evidence of biological breakdown. The Japanese aesthetic concept of mono no aware, which finds beauty in the transient nature of life, and similar cultural recognitions of decay's role in natural cycles demonstrate how decomposition chemistry connects with human philosophical and emotional responses to temporal change. The microscopic beauty of decomposition processes, visible through careful observation of fungal networks, bacterial colonies, and the intricate patterns of enzymatic breakdown, reveals the hidden complexity and elegance of decay chemistry. The delicate structures of mold growing on organic matter, the crystalline formations of mineral precipitates in decaying wood, and the rich colors produced by various decomposition products all demonstrate how the chemistry of decay creates its own forms of beauty. The eternal cycle of transformation. In the recognition that all chemical change represents part of endless cycles of molecular rearrangement and elemental circulation, time reveals itself as the conductor of a cosmic symphony where atoms and molecules dance through countless configurations while never being created or destroyed, only transformed from one chemical arrangement to another in patterns that span scales from molecular to galactic and timescales from femtoseconds to cosmic ages. The conservation of mass that governs all chemical transformations ensures that the atoms participating in rust formation, fruit ripening, color fading, and all other temporal chemistry have been cycling through different molecular configurations for billions of years, having been forged in stellar cores, distributed through space by supernovas, and incorporated into countless chemical compounds throughout the history of the solar system and the evolution of life on Earth. The thermodynamic principles that drive chemical change operate through the universal tendency of systems to evolve towards states of lower energy and higher entropy, creating the directionality of time that distinguishes past from future, while ensuring that all chemical processes eventually reach equilibrium conditions where forward and reverse reaction rates balance and no further net change occurs, without external energy input or environmental modification. The catalytic processes that accelerate chemical transformations without being consumed demonstrate how nature uses specialized molecules to lower activation barriers and enable reactions that would otherwise occur too slowly to be observed. From the enzyme catalysis that drives biological chemistry to the metal catalysis that enables industrial chemical production and the atmospheric photochemistry that maintains global chemical cycles. The coupling of chemical reactions that allows energy-releasing processes to drive energy-requiring transformations creates the complex reaction networks that characterize living systems, where the oxidation of food molecules provides the energy needed for protein synthesis, muscle contraction, neural activity, and other biological processes that maintain the organized complexity of life against the general tendency toward molecular randomization. The information storage and processing capabilities of chemical systems, from the genetic information encoded in DNA sequences to the catalytic information embedded in enzyme structures, demonstrate how chemistry can create and maintain complex patterns of organization that persist through time while enabling the replication and evolution of these patterns through chemical processes that operate with extraordinary fidelity and occasional beneficial mutations. The emergence of complexity from simple chemical components illustrates how time and chemistry collaborate to create structures and processes that transcend the properties of their individual molecular constituents, with examples ranging from the self-assembly of cellular membranes from simple lipid molecules to the formation of complex ecosystems from the chemical interactions between diverse organisms and their environment. The global biogeochemical cycles that circulate elements like carbon, nitrogen, phosphorus, and sulfur through living organisms, the atmosphere, the oceans, and the Earth's crust demonstrate how local chemical processes connect to form planet-scale systems that have maintained the chemical conditions necessary for life throughout Earth's history while adapting to changing environmental conditions and biological evolution. The chemical basis of consciousness and awareness represents perhaps the most remarkable demonstration of time's creative power, where the complex neural chemistry that enables perception, memory, emotion, and thought emerges from the coordinated activity of billions of neurons using the same basic chemical principles that govern all biological processes, but organized into networks of extraordinary complexity and sophistication. The technological applications of temporal chemistry, from the controlled release of pharmaceutical compounds to the optimization of industrial reaction conditions to the development of materials that change properties over time, demonstrate humanity's growing ability to design and direct chemical processes for specific purposes while working within the fundamental constraints imposed by thermodynamics and reaction kinetics. The aesthetic appreciation of chemical change encompasses recognition of the beauty inherent in molecular transformation, from the elegant symmetries of crystal structures to the flowing patterns of reaction diffusion systems, to the rich colors and textures that emerge when chemistry and time collaborate to create visual complexity from simple starting materials and reaction conditions. The philosophical implications of understanding chemical change include recognition that consciousness itself participates in the ongoing chemical evolution of the universe, that human awareness and intention can influence chemical processes through technology and choice, and that the chemical basis of thought and emotion connects human experience with the fundamental processes that govern all matter and energy throughout the cosmos. The temporal perspective that emerges from contemplating chemical change reveals that what appears as decay or destruction on human timescales often represents construction and creation on longer timescales, with the decomposition of individual organisms enabling ecosystem renewal, the weathering of rocks creating soil for new plant growth, and the fading of colors allowing new chromatic relationships to emerge. The unity that underlies all chemical diversity becomes apparent through recognition that different chemical phenomena represent variations on common themes, with the same fundamental principles governing rust formation and memory consolidation, fruit ripening and star formation, color fading and consciousness evolution, demonstrating how time uses chemistry as a universal language for transformation and creation throughout the universe. Now, rest in the recognition of this eternal chemical dance, feeling how consciousness participates in the same molecular transformations that shape rocks and ripen fruit, sensing the deep unity that connects all chemistry through the patient work of time, drifting into the peaceful awareness that all change represents aspects of one continuous process of cosmic evolution. The silence of molecular dreams. As awareness settles into the deepest contemplation of time's chemical artistry, there emerges a recognition that beneath all the molecular transformations and atomic rearrangements lies a profound stillness, not the absence of change, but the unchanging field of awareness within which all chemical processes arise and dissolve, like waves on an infinite ocean of pure possibility. This molecular silence represents the space between transformations, the pause between reactions, the eternal present within which all temporal chemistry unfolds. The atoms that participate in every chemical reaction carry within their electronic orbitals the quantum mechanical waves that represent pure potentiality, existing in superposition states until the moment of chemical interaction collapses these probability distributions into definite molecular configurations. This quantum foundation of chemistry reveals that even at the most fundamental level, matter exists in dialogue with consciousness, with observation and interaction determining which of many possible chemical outcomes actually manifests in the physical world. The chemical bonds that connect atoms into molecules exist not as static links, but as dynamic probability clouds, where electrons dance in patterns that defy classical description, while creating the stable relationships that allow complex molecular structures to persist through time. The contemplation of chemical bonding reveals the mysterious nature of the forces that hold matter together while allowing it to transform, creating the paradox of persistent structure within constant change. The kinetic energy of molecular motion creates the thermal background against which all chemical reactions occur, with atoms and molecules in constant motion, even at absolute zero, due to quantum mechanical zero-point energy. that ensures no chemical system ever reaches complete stillness. This perpetual molecular motion provides the energy needed for chemical transformations, while creating the dynamic environment within which chemical equilibrium represents not stasis but balanced activity. The activation barriers that must be overcome for chemical reactions to proceed create temporary molecular configurations that exist only briefly before transforming into products or reverting to reactants, demonstrating how chemistry operates through transient states that serve as bridges between different molecular arrangements. These transition states represent chemistry's exploration of configurational space as molecules seek new arrangements of lower energy. The catalytic cycles that accelerate chemical transformations operate through the temporary formation of intermediate complexes that lower activation barriers while remaining unchanged at the end of each catalytic cycle, demonstrating how chemistry can create self-renewing processes that facilitate change while maintaining their own structural integrity. The beauty of catalysis lies in this combination of enabling transformation while preserving identity. The spontaneous symmetry breaking that occurs in many chemical systems, from the formation of chiral molecules to the development of concentration gradients, demonstrates how chemistry can create order from disorder through processes that appear random individually but produce organized patterns when viewed collectively. This emergence of pattern from chaos represents one of chemistry's most profound creative capacities. The chemical basis of biological rhythms, from circadian cycles to heartbeats to brain waves, reveals how chemistry creates temporal patterns that organize life processes according to periodic schedules that synchronize internal biological chemistry with external environmental cycles. These chemical clocks demonstrate how molecular processes can measure and respond to time while participating in its creation through rhythmic activity. The memory effects in chemical systems, where past chemical history influences present chemical behavior, demonstrate how molecules can store information about previous experiences while remaining available for new transformations. The hysteresis effects in complex chemical networks create chemical memory that allows systems to learn from experience while maintaining flexibility for adaptation to changing conditions. The chemical communication networks that operate within and between living organisms create information processing systems of extraordinary complexity and sensitivity, where molecular signals carry detailed information about internal states and external conditions while triggering appropriate chemical responses. These chemical communication systems demonstrate how chemistry can create intelligence through molecular information processing. The chemical creativity that manifests in biological evolution, where random molecular variations create new chemical capabilities that may prove advantageous under changing environmental conditions, demonstrates how chemistry can explore new possibilities through trial and error processes that operate over extended time periods. The generation of chemical diversity through mutation and selection represents chemistry's method for discovering new solutions to environmental challenges. The chemical basis of consciousness remains one of science's greatest mysteries, involving the emergence of subjective experience from the objective chemistry of neural networks through mechanisms that may require new understanding of how chemistry and consciousness interact at fundamental levels. The hard problem of consciousness may ultimately require recognition that awareness and chemistry are more intimately connected than current scientific paradigms suggest. The aesthetic dimension of chemical beauty encompasses not only the visual appeal of chemical phenomena, but the deeper recognition that chemistry creates beauty through the elegant mathematical relationships that govern molecular behavior, the symmetries that characterize chemical structures, and the harmonious patterns that emerge when chemical systems reach optimal configurations through natural selection or designed optimization. The spiritual implications of chemical understanding include recognition that the same molecular processes that create and maintain biological life also generate the neural activity that supports consciousness, awareness, and the capacity for spiritual experience, suggesting that the material and spiritual aspects of existence may represent different perspectives on unified chemical processes rather than separate domains of reality. The ultimate mystery of chemical existence lies in the question of why matter possesses the capacity for such extraordinary organizational complexity and creative potential, why atoms can combine to form molecules capable of consciousness, memory, emotion, and spiritual awareness, and how chemistry can serve as the bridge between the quantum mechanical foundations of matter and the emergence of mind and awareness in complex systems. The peace that emerges from contemplating chemistry's creative potential comes from recognizing that consciousness participates in the same fundamental processes that govern all molecular transformation, that awareness itself may represent chemistry's highest achievement in organizational complexity, and that the chemical basis of thought and feeling connects human experience with the cosmic creativity that operates throughout the universe. The eternal quality of chemical law demonstrates that while specific molecular configurations arise and dissolve through time, the principles that govern chemical behavior represent timeless truths that have operated since the formation of the first atoms and will continue to operate throughout the future evolution of the universe, providing stable foundations for endless creative exploration of chemical possibility. Now, rest in this molecular silence, feeling the deep peace that underlies all chemical transformation, sensing the unchanging awareness within which atoms dance and molecules transform, drifting into the recognition that consciousness itself represents chemistry's most beautiful creation, where awareness and matter merge in the eternal stillness from which all change emerges. Sleep now in the gentle rhythm of molecular motion, where electrons orbit in quantum clouds of possibility, where chemical bonds form and dissolve in patterns of elegant complexity, where the chemistry of consciousness creates the awareness that contemplates its own molecular foundations in the infinite silence that contains all transformation, the eternal peace that supports all change, the unchanging love that creates and sustains all chemistry throughout the cosmos. In this deepest rest, where awareness merges with the quantum foundations of matter, where consciousness recognizes itself in every molecular transformation, where the chemistry of time reveals itself as the chemistry of timeless awareness, sleep comes as a return to the source, not as unconsciousness, but as the deepest recognition that all chemistry participates in one infinite process of creative evolution, forever exploring new possibilities for beauty, complexity, and conscious awareness, in the eternal dance of atoms and consciousness, forever and ever and ever. Amen.